Hello, yes, my voice is audible. Please let me know.
जहाँ भाई भाई है इनमें लड़ाई करवाने का कोई मकसद नहीं बनता क्योंकि लंगरा करके बहुत सारे गरीबों को
Hello, my
आसरा
voice
मिल
is
गया
audible
है।
to all of you.
दिल्ली ने बॉर्डर के ऊपर पुख्ता प्रबंध ਜਿਹੜੇ ਕੀਤੇ ਗਏ ਨੇ ਇਹਨਾਂ ਦੇ ਵਿੱਚ ਤੁਸੀਂ ਦੇਖ ਸਕਦੇ ਹੋ ਹਰ ਵਿਅਕਤੀ ਆਪਣੀ ਸੇਵਾ ਨਿਭਾ ਰਿਹਾ ਹੈ ਹਰ ਇਨਸਾਨ ਜਿਹੜਾ ਉਹ ਲੰਗਰ ਛਕ ਰਿਹਾ ਹੈ ਕੋਈ ਭੇਦਭਾਵ ਨਹੀਂ ਕੀਤਾ ਜਾ ਰਿਹਾ ਤੇ
Hello,
ਕੋਸ਼ਿਸ਼ ਇਹ
yes,
ਕੀਤੀ
my
ਜਾ
voice
ਰਹੀ
is
ਹੈ ਕਿ
audible.
ਅਸੀਂ
Please let me know. Are
ਜਿੰਨੀ
you able
ਕੋਸ਼ਿਸ਼
to
ਕਰੀਏ
listen to me?
ਕਿਸਾਨਾਂ ਦੇ ਹੱਕ ਦੇ ਵਿੱਚ ਨਿਕਲੇ ਤੇ ਐਕਚੁਅਲੀ ਕਾਲੇ ਕਾਨੂੰਨ ਜਿਹੜੇ ਨੇ ਕਿਸਾਨਾਂ ਤੇ ਥੋਪੇ ਜਾ ਰਹੇ ਨੇ ਉਹ ਕਿਸੇ ਨਾ ਕਿਸੇ ਤਰੀਕੇ
So, is it clear to now, all of you? Is it clear to all of you? Please let me know. Okay. So, good evening to all of you. Sorry for the network issues before. I think it is okay now. Shubhjeet, please, if possible, take your earphones along with you. It will be okay soon. The noise may be because of these our fan in the laptop. Maybe because of it there is a noise. But don't worry. Just put your earphones if possible. Okay. So just put your earphones along with you. Okay, so now I have to start our. Yes, I understand there is some noise. It will be okay soon. The noise is because of this laptop. So now I am just going to start our today's lecture because we are already very late. We have to start it, but now it will be okay soon. No issues, Purvati. It will be okay soon. No issues. Just, just let me know about something regarding your review paper. Are you doing it? Everything is fine. Please let me know. Everything is fine. The review paper and something, is it fine or not? So that I will be able to start the session because we are already very late. I have my next session, my next lecture also. So that is why I have to do it as soon as possible. Okay, so now just I am going to share today's lecture which I have to present. My voice is audible to all. Please let me know my voice is audible. So I have to start today's session in which we have to do types of gene action. You have already done Mendel's law before. So I have to start with types of gene action. So I am going to share the presentation. So I have to share the PPT with all of you. Just wait for a while. And yes, tomorrow you all have a holiday, so we will take a class for extra hours so that we will be able to do as much work as possible. So I will take little bit long sessions tomorrow. Tomorrow it will be a little bit long session, so be ready with it. Getting my point? So tomorrow the sessions will be little bit long. Yeah. Okay. Okay, good. So tomorrow and on weekends, that is on Saturdays and Sundays, I will try to take the sessions little bit long. So I have to start today's lecture with gene interactions. Basically, what we have to do in it, I will show you. Let me. Yes, I will start from here. So you all have done the Mendel's law, the Mendel's ratios which are there. So now we have to start interaction of genes, modification of dihybrid ratio, and extension of Mendelian genetics. So we have to start interaction of genes, that means the interaction within the genes, modification of the dihybrid ratio which is given by Mendel, and then some extension by Mendelian genetics, some extensions of Mendelian genetics. Getting my point? So yes, Mendel performed monohybrid and dihybrid cross taking sweet pea as an experimental plant. You all know about it that Mendel takes sweet pea as a plant for his experiments, and he performed monohybrid as well as dihybrid crosses. So in monohybrid cross, only one trait and its two alternative forms were taken at a particular time. So in monohybrid, only one trait. Suppose Mendel has taken the color of the flower. So only one character is taken by the Mendel in monohybrid cross, and it has only two alternative forms. Like suppose the height of the tree or height of the plant, it may be either tall or dwarf. So only one character is taken, and with only two alternative forms of that particular character at a particular time. So if we are just discussing about the flower color, so it will be either a red flower or a white flower. So only two alternative forms are there in case of Mendel in only a monohybrid cross, when we have to work only with a single character. So this is the monohybrid cross. So in the monohybrid cross, there are three possible genotypes. The genotypes will be capital R, capital R, capital R, and small r. That means the genotypes will be either red, either white, or the dominant one, which is the red one, with phenotypic ratio 3 is to 1 and genotypic ratio 1 is to 2 is to 1. So you have to keep this thing in mind that in Mendel's law, during monohybrid cross, only single character is taken with two alternative forms, and they have phenotypic ratio 3 is to 1 and genotypic ratio 1 is to 2 is to 1. Okay. So in dihybrid cross, two characters or traits were taken at a particular time. So two characters like height of the plant, that means it is either tall or dwarf, and color of flower. So tall, red, tall, white, dwarf, red, dwarf, white. So in dihybrid cross, the phenotypic ratio is 9 is to 3 is to 1. Getting my point? In case of monohybrid, the ratio is 3 is to 1. In case of dihybrid, the ratio is 9 is to 3 is to 1. Please let me check whether you are able to listen to me. So are you able to get it what I want to tell you all? Are you able to listen to me properly? So let me know about Mendelian's monohybrid ratio, or you can say Mendelian's phenotypic ratio. Any one of you, please give me the answer. The phenotypic ratio which is given by Mendel. And all of you have to answer the phenotypic ratio which is given by Mendel. The phenotypic ratio for the monohybrid cross, or what is the difference between monohybrid and dihybrid cross? What is the difference between a monohybrid and dihybrid cross? What is the difference between monohybrid and dihybrid cross? What is the phenotypic ratio of monohybrid cross and phenotypic ratio of dihybrid cross? Okay, good, all of you. Please let me know the difference between monohybrid and dihybrid. Yes, very good, Nidhi. We have Urja, we have Pooja, Pratyasha, Abha, Komal. Yes, very good. Anvesha Mandal, very good. No, Pooja, what is dihybrid? Basically, in dihybrid cross, we are taking two characters together, or you can say two traits together. Yes, good, Nidhi, good. Yes. So, When we are working with dihybrid cross, we are taking two characters together, and in case of monohybrid, we are taking a single character with only two alternative forms, either tall or dwarf. The color may be either red or white. Okay, the size may be rounded or wrinkled. So these only a single character is studied. So all of this is up to this is all things are related to Mendel. But now we have to discuss the interaction which is there in between the genes. So now we have to, these are the ratios which are given by Mendel, but now we have to focus on some gene interactions which is there in between the genes. So the phenotypic ratio in case of dihybrid cross is 9 is to 3 is to 3 is to 1. Okay. So this is the thing which is given by Mendel. So the phenotypic ratio is 9 is to 3 is to 1. So this is the dihybrid cross. This is the cross how we are able to take two traits or two characters together, like tall and red color, dwarf and white flower or white color. So in the F1 generation we are able to get T, capital T, small t, capital R, small r. That means tall plants with red color, because capital is tall and capital R is red. So we are able to get tall plants with red flowers. So by doing the possible combinations, we are able to get the ratio which is 9 is to 3 is to 3 is to 1. Okay, I hope all of you must know about this cross. So this is, you have to keep in mind the ratio only. So by seeing Mendel's dihybrid ratio, it becomes clear that different characters are inherited independently. Because of that, four combinations are produced in F2 generation. So this is the Mendel's conclusion. Mendel concluded on the basis of results which he performed on sweet pea plant. Number one character or one trait is controlled by a gene existing in two alternative forms. So, according to him, a character is controlled. Suppose the character is height. So, height is controlled by a particular gene, or you can say factor, existing in two alternative forms. So, height may be either tall or dwarf, or you can say another character like color. So, the color may be either red or white, and it is again controlled by a single gene or factor. This is according to Mendel. This is according to Mendel that a character which may be height, which may be color, which may be size is controlled by a particular gene, number one, and that particular character has two alternative forms only, either tall or dwarf, either red or white, either round or wrinkled. This is the observation which was given by Mendel. Number two, that different traits assorted independently. Different traits assorted independently. Segregation of two genes is independent of each other. According to it, that the characters like tall and the red color, they are independent of each other. Getting my point? The characters are independent of each other. The height character is independent of the color character. So you have to remember these two observations by Mendel. Okay, but now after Mendel, there are so many scientists which put another observations, or you can say another theories. Up to it, is it clear to all of you? Are you able to understand Mendel's conclusion? Please let me know. Then I will start the another scientist observations. Up to this, is it clear to all of you? Are you able to get the conclusion which was given by Mendel? Please let me know. Are you able to get the conclusion which was given by Mendel? All of you, please let me know. So, Mendel has given us two conclusions. The one is that the character is controlled by single gene and each character has two alternative forms only. Number two, that the two characters, they are independent of each other. Okay, getting my point. So this is the observation which was given by Mendel. So now I will show you the observations by another scientist. Getting my point. So now we will keep forward. Okay, okay, good, good. So you are getting it. But after it, there are so many scientists. So after Mendel's work, when Mendel's observation, there are some scientists like Hugo de Varies, Carl Correns, and Teschmark. So after Mendel, Hugo de Varies, Carl Correns, and Teschmark, they repeated the same pattern of work using other organisms. So instead of pea plant, they worked on another organisms and different phenotypes. But the results were not as always according to Mendel's law of inheritance. So when the scientists they take another organisms instead of pea plant and they take another character, they found the results totally different. Okay, so the results were not always according to Mendel's law of inheritance. According to Mendel, the laws which are given by Mendel is not appropriate on all organisms. So, law of dominance and law of independent assortment, these two laws not work on all organisms. Getting my point? So it became apparent that it is not necessary that a single character like height or color or shape is controlled by a single gene only. No, it is not like that, that tallness is characterized by this tallness or height is controlled by this particular gene. It is not possible. So it is not true that a gene has only two alleles, may have many alleles, and the phenotype may be result of interaction of two genes. So, according to Mendel, one gene, one character, it is not like that. So, a gene has only two alleles, it is not possible. It may have many alleles, or you can say many characters, or many, instead of tall and dwarf, it may have intermediate also. Like instead of red color and white color, the color may be pink also. And the phenotype may be the result of interaction of two genes and their alleles or a character may be controlled by more than one gene. So according to Mendel, single character is controlled by a single gene. This is not possible. And according to Mendel, a single character has two alternative forms only. So height has either tall or dwarf. Color will be red or white, but it is not like that. So instead of two alleles, a gene may have many alleles. So in such a condition, two or more genes may interact. The resulting phenotype may be the result of interactions. So when we are saying gene interactions, it means instead of a single gene, more than two genes are interacting. So the result which we get, it is the interaction of more than two genes. Are you able to get it right now? Please let me know, is it clear to all of you? Is it clear to all of you? Please let me know. Up to now, is it clear to all of you? Please let me know. hope it is clear to all of you. My voice is audible to all of you. So up to now, is it clear to all of you? Please let me know my voice is audible. So that I will continue further. Please let me know. Okay, okay, good. So interaction is basically when we are discussing, when we are taking more than two genes, then it has an interaction. So in when more than two genes interact, the resulting phenotype may be the result of that particular interactions. So now I am just going to let you know about how the gene interacts, how the phenotype. So now there are two things. We have phenotypic ratio and we have genotypic ratio. Now what is the phenotypic ratio basically? Like in monohybrid cross we have 3:1. Na? So what is phenotypic ratio? When we are discussing the physical appearance, that how physically they are looking, like the height, the color. So suppose gene A is responsible for red color. Suppose there is a one gene which is responsible for red color. Gene B is responsible for white color. So when both genes, both A and B are present, together they may give rise to new color which is pink. According to according to Mendel, when red color and white color, or red plant and white plant, they are hybrids, it results into the red color, which is law of dominance. But here they are saying when one gene is interacting, the red color, and the gene B which produces the phenotype white, so when both A and B are present, they may give rise to new phenotype which is pink color. Okay, but these genes still obey independent assortment pattern. What is independent assortment? That they are just they are independently assorted. They are not dependent on each other. So if A and B are two genes, both dominant over the recessive alleles, that means here it should be capital B basically. Okay, here it is capital B. So the interaction will depend on the presence of both dominant alleles A and B and absence of A and B. So Mendel was fortunate to get the ratio of 3:1 and 9:3:3:1 in case of pea plant. But let me conclude it. The traits which has been taken by Mendel has only one pair of alleles of a particular gene. So for the height, the alleles are only two, that is capital T and small t. But it is not possible in all organisms or in all experiments. But in case of Mendel, he was lucky that he was able to select those characters or those genes which has only two alleles, either capital T or small t. So, the genes and their alleles for different characters were located on different homologous pairs of chromosomes, or even if on the same homologous pair of chromosomes or homologous pair of the genes for different traits were present at a distance sufficient for crossover. I will let you know about this later on, because this is a little bit related to linkage, the topic which we have to cover later on. So I will not discuss this point at that time. But yes, I will let you know that Mendel was quite lucky. The characters or the genes which he has taken has only two alleles or one pair of alleles, either height, either capital T or small t. So this is the brief introduction of interaction of genes. But before this interaction, you must know about Mendel or the variations which are there in case of Mendel's law. Getting my point? So now we have to discuss the interactions which are there. So genes usually function or express themselves singly or individually. Generally it happens that genes, they generally express themselves individually. But sometimes what happens when there are more than two genes, so where two genes of same allelic pair or genes of two or more different allelic pairs influence one another. This is called gene interaction. So when gene A is present alone, when gene A, it's only example, when gene A is present alone, the phenotypic appearance is different. Suppose when gene A is there, it gives us red color. Gene B gives us white color. But when both the genes, or you can say pair of genes, or where two genes, they interact with each other. That means when gene A and gene B are together, instead of giving red or white color, it gives us pink color. I hope you are able to get me properly. I don't want to make it, hodgepodge, or you will feel confused. Not at all. I am just saying, according to Mendel, when gene A and gene B interact, only the dominant one will show its phenotypic character. That means if R and red and white, they will interact, only red will show its character. But here in this case, in case of dihybrid, we are saying that when gene A and gene B interact, gene A usually gives red color, gene B usually gives white color. So, but when gene A and B are together, they give us the third color, which is the pink color. So, genes usually function or express themselves singly or individually, but many cases are known where two genes at the same allelic pair or genes of two or more different allelic pairs influence one another. This is called gene interaction. This is the allelic pair of interaction. We have another interaction which is known as non-allelic gene interaction. Non-allelic gene interaction. Now what happened in this? What happened? These are interactions between genes located on the same chromosome or on different but homologous chromosomes controlling a single phenotype to produce a different expression. So I will give you examples to let you understand this non-allelic gene interaction. Here basically what happens, there is an interaction between the genes. There is an interaction between a genes for controlling a single phenotype. Here we are just working on a single phenotype to produce a different expression. So each interaction is typically in itself the ratio obtained from those of the Mendelian dihybrid ratio. So there will be a little bit variation from the Mendel's dihybrid ratio, which is 9:3:3:1. So these are the interactions which we have to study in detail. This is the interaction which is given by Mendel, 9:3:3:1 in case of dihybrid cross. But we have to study these interactions, 9:3:4. 12 is to 3 is to 1, 9 is to 7. So we have to study epistasis, we have to study complementary genes, supplementary genes, duplicate genes, inhibitory genes, and polymeric genes. We have to study all these interactions which are there in between the genes, and all these interactions have some effect on the phenotypic ratio as well as on the genotypic ratio. So let me check whether you are getting me or not. So up to it, is it clear to all of you or not? Up to it, is it clear to all of you or not? Please let me know. I am just giving you a brief introduction only. We have to discuss all these things in very detail. Yes, I know about codominance, Shivani. Don't worry about it. I know about it. But I just want to clear you something related to Mendel's and something which is, the work which has been done by the scientists later after the Mendel. So up to now, is it clear to all of you? Please let me know. All of you, up to now, is it clear to you or not? All of you, are you able to get it? Please let me know. Otherwise, I will ask you questions. Then you have to answer those questions also. Please, all of you. So this is the very basic things of genetics. You, all of you must be, you have to understand all these things very clearly. There should not be any doubt regarding Mendel's law, what is proposed by Mendel, why Mendel is lucky, why he is able to, take only the gene with only two alleles. So there are some things which you have to know initially. Initially. Did you know Mendel's finding, the finding he provided to all of us, is not recognized, was not recognized when he was alive. His work was not appreciated when he was alive. He was not appreciated at all at that time when he was performing these experiments and collecting the data and everything. His work was not appreciated and he was declared as, you know, like mental or something you can say. So his work was not appreciated during his whole life, and his work was appreciated very, after his death, lots of years back then, by some another scientist who took his data and just doing the things, and that scientist was able to find that Mendel was totally right. But the persons which are there, who were working along with Mendel, or the, the society, they were not ready to accept what Mendel was proposing. So yes, there is something. This is life. When you are right, no one is there. Sometimes no one is there to take it positively. Okay. So I just want to let you know that this is the story of Mendel's work. He was known as father. He is known as, not was. He is still now known as father of genetics, but he himself was not aware of it. Yes, Pratyasha Mishra, I will let you know about codominance. Just keep some patience. I don't know why you become so impatient. I am just here right now on Mendel, and then you jump on codominance. If you have some knowledge, please keep this knowledge along with you. But please be some patient also, because I have to think all of you. Some students are totally new. Some students are totally new to Mendel also. So keep some patience. Right now I am on Mendel, and you just jump to codominance. Tomorrow I will be on codominance. You will jump to blood group or multiple alleles. Please keep some patience. I request all of you. Please. Because genetics is a very these things are very new to some students, and I want to keep the interest, you know, keep growing in all those students, because they are from different batches, from different streams. I don't want to I don't want that they lose their interest in genetics. So please pardon me right now if I am not able to, clarify you. So this is the thing which we have discussed today. We have discussed Mendel's law, and we have discussed that so we have just discussed Mendel's law, the things which Mendel was able to tell us, and the things which are not as appropriate as Mendel was, you know, letting us know. So this is the one thing. And yes, I have one more question for all of you, which you have to tell me tomorrow, not today, but tomorrow, that it is a homework for all of you. You have to tell me that why Mendel is, Mendel's work is considering, or Mendel is considering as lucky enough. How he was lucky, how he was able to do the experiments, and his whole work is taken as completely right. So you have to tell it to me tomorrow, or I will wait your answers in the Telegram group also. It is especially for the newcomers. I want the newcomers to search lots of things related to Mendel and let me know why Mendel was considered lucky, or for his all experiments or for his all observation, why he was considering as a lucky man. Okay, so this is your homework. You have to let me know. And yes, we have these interactions. We will just start a little bit interaction today. But before it, I also want to say you one more thing. Let me 1 minute, please. Before it, I just want to ask you one more thing. Tomorrow is Saturday. I know all of you are free. So instead of taking this class at 7 p.m. or 7:45 p.m., I will take the class early in the morning around 11 a.m. and I will take it for 2 hours long session because you already know your ma'am is not here right now along with us. So I have to complete the syllabus as much as I can, and I have my own session just right after your class. So on Saturdays and Sundays we will take the long classes, okay? Up to 2 hours it will be there, so be ready with it. And tomorrow I will take, but Himangi, tomorrow is Saturday. If you have your lecture in college, then you please go through with the YouTube recording, because for all my batches, I have my biochemistry batch also. On Saturdays and Sundays specifically, I take 2 hours long session. Okay, so if you are unable to join the class, you can just go with the link, and you know you can go through by the link by your own self, and you will be free. But tomorrow is Saturday. Okay, then we will take it from 3 p.m. to 5 p.m. But I will not, or 1 to 3 p.m. I will try to do it 1 to 3 p.m. That means after lunch I will take. You can do your classes before also, but I will take it after. You can say from 3 p.m. to 5 p.m. Okay. And one more thing, for Sunday it will be from 12 or from 11 or from 12 p.m. So be ready with it. At that time I will not listen 12 or from 11 or from 12 p.m. So be ready with it. At that time I will not listen to any excuse, because I have my another class also right after your class. So I will not take it in the evening. And be ready for 2 hours long session also. So if you have any problem, you can take the session, recorded session, and you can listen it to later on. Otherwise, we will try to take it just after the lunch. By 3 to 5 p.m., I think you all will be comfortable. Okay? No, no, not after 5 p.m. By 3 to 5 p.m., I will take your session. Otherwise, you will just go through the recorded video. No worries, dear. If you will not be able to take the classes, because of your online class, you will not be able to take the class, YouTube class. No worries. You will be get the recorded video. You can watch it anytime when you will be free. Because please try to understand, your memory is not there, and I am here only for you to complete your syllabus. So please cooperate with me, because I have my own schedule also. That is why, okay, so please don't worry, don't panic, no issues at all. You will get the recorded lectures, and you can watch it anytime when you will be free. Or if you have any query or any question, you can message me on Telegram. I will definitely reply you, or we will chat on Google Meet also. So no issues at all, dear. Not at all. Yes, yes, please, please. Thank you all for the cooperation. So maybe I will take it from 3 p.m. or by 4 p.m. But it will not be late by after 4 p.m. 4 to 6 I will take it. Otherwise my schedule is very busy. So I have to think of all the students which are in touch with me. So please allow me the same. And yes, we have to complete these things which I want to share with you. Like we have to complete tomorrow, we have to do types of gene action. All these interactions we have to discuss it in detail. Then we will have to do quantitative traits and qualitative traits and multiple alleles. So topic number 2 and 3 will be covered tomorrow. A- and on Sunday we will try to complete the coming topics. Okay. So that is all which I want to discuss with all of you. Now, if you have any query regarding the review paper, please let me know. If you have any query regarding the review paper, please let me know. Have you start writing your review paper? Please, okay. I didn't send you the topics in the group. Okay, I will send it just after this lecture will be closed. I will send you the topics. But the topics which I will send you, they will be related to genetics only. Okay, Himangi, I will send the topics, but they will be related to genetics only. Okay, I will just right now I will just send it in a Telegram group. Maybe I forget before. So, but they will be related to genetics only. All of you, please keep this in mind. So I'm just putting it in your group so that you will be able to select any topic of your own point of interest. Okay. So you can select any topic from these topics, or you can do it by your own self also. And just please be there in touch with me so that I will able to help all of you. No, John, you have to learn the you have to read the review papers only to write your own particular review paper. You have to read those review papers in detail, or you can also read research papers. No issues at all. The only work you have to do, you have to compile their work together into your own review paper. So you can search either for the research papers or also for the review papers. No issues at all. You can take both research paper as well as review paper. If you want to write it in a group, you can select any single topic, but only 5 students are allowed to write a review paper together on a particular topic. Getting my point? So if you want to write a review paper in group on a single topic, only 5 students are allowed. Yes, Lopa Mudra. Yes, you can take any topic of your own point of interest, no issues at all. Yes, John, you have to read both. You are free to read anything, either research papers, review papers, case studies, everything. All of you have to read, and you have to search for good content and make your own review paper. Getting my point? Okay, so I think that's all for today. I have my another lecture series by 5 or 10 minutes. So that's all for today. Just go with these. Tomorrow we will take 2 hours long session. So be ready with it. Be prepared with it because we have to cover two important topics tomorrow: the gene interactions as well as multiple alleles. So be there. Don't worry about the last date. Just focus on the topic and let me know. And yes, we will meet tomorrow. Till then, take care of yourself. Till then, take care of yourself. Good night to all of you, and hope we will meet tomorrow on proper timing. Okay, so don't be late and be ready for tomorrow's early session. Yes, you can collaborate, no issues at all, but be ready to do something regarding the review paper. By 10th of December, you have to send me your abstract along with the title or the topic of your own selection. So please, you have ample of time and do something. And yes, I want to say thank you all because you just do your homework and you have sent me the no, not you. That one the students from biochemistry. So you have your homework along with you and you have to answer me the question either in telegram or even tomorrow also. So I will ask you a question again. So you have to answer that question particularly. Yes. You can take any topic. Don't repeat the same question. I allowed all of you to select any topic of your own point of interest. So good night, bye, take care. Wish you all the best for your review paper. Hope we will be able to publish it in a good journal. So take care all. Please all of you take care of yourself. Bye. So be ready tomorrow before the long hour session. All of you please be ready. Okay. Bye, Jyoti, Archana, Mangi, Sagarika, Pratyasha, Nisha, Komal, Nidhi. Take care of yourself, all of you. Bye. Anvesha. I'm sorry if I pronounced your name wrong. Please pardon me. Maybe I just pronounced your name wrong. Anvesha Mandal. Shivani, please don't answer right now. Please allow all of them to search, otherwise they will copy you. Please. Otherwise you are right. I know you are right, but let them search something. Then we will discuss this thing tomorrow also. Yes, he was lucky because the characters were not linked together and they are independently sorted. But we are waiting the positive response from all of you till tomorrow. So take care. Bye. Good night. We will meet soon. Bye. Okay, take care.